These mice are identical twins. They don't look identical, but they have identical DNA.
Wait, I thought hair color was determined by your genes.
It is. These mice have the same genetic sequence, but the way these genes are expressed is different. Let's explain gene expression. I'll review how the mice have different color fur at the end of the video. Gene expression is the process of turning the genotype into the phenotype.
What's genotype and what's phenotype?
The genotype is all of an organism's genetic information, while the phenotype is the set of observable characteristics like hair color and eye color. For example, gene expression is how the gene for brown eyes results in actually having brown eyes.
Okay.
During gene expression, the genes undergo transcription and translation. Transcription is producing a strand of mRNA using DNA as a template. Translation is using the mRNA to produce a specific protein. In the case of eye color, the protein catalyzes the production of pigments for the eye, which gives it its color.
Wait, so all phenotypes like height and hair color are produced by this process?
Most of them, yeah. In any cell, the full set of genetic information is a genome. The full set of mRNAs transcribed is a transcriptome, and the set of proteins produced is a proteome. Here's a question for you to think about. The genome is identical in almost all your cells, so how can they produce different sets of proteins? This is why we need regulation of gene expression. We will be talking about regulation by proteins, regulation by degradation of mRNA, and regulation by epigenetics. Let's start with regulation by proteins. This is a strand of DNA. Here's the gene. For a gene to be expressed, RNA polymerase needs to transcribe the DNA into mRNA. To do so, it binds to the promoter, a section of the DNA that is located before the gene.
What's the Tata box?
It's a sequence in the promoter that has lots of A and T bases. Those are 2 of the 4 DNA bases. Many promoters in eukaryotes have it. To clarify, eukaryotes are organisms with cells that have nuclei, like animals.
Okay, but why is it there?
This allows a general transcription factor to bind to it, so RNA polymerase attaches in the right place.
Oh, what's a transcription factor?
Good question. A transcription factor is a protein that regulates transcription by binding to the DNA. Some, like the one I just explained, are general transcription factors. Other transcription factors are repressors, meaning they bind to the silencer sequence. As the name indicates, this decreases the rate of transcription. We also have activators, meaning they bind to the enhancer sequence. This increases the rate of transcription. Let's give a classic example of how these interact. The lac operon. This is a system in which a type of bacteria called Escherichia coli or E. coli regulates the metabolism of lactose. Basically, how they use lactose as their food. To digest lactose, they need to produce an enzyme, but this process requires energy. Therefore, E. coli bacteria have a system that allows them to produce the enzyme only when there is enough lactose to save energy. Cool. Normally, there is a repressor bound to the DNA, blocking transcription. However, when lactose is present, it binds to the repressor, which deactivates the repressor, allowing transcription to occur.
Oh, and when there isn't lactose, the repressor binds to the DNA again, so transcription is blocked. Oh.
Yep, you got it. The next type of gene regulation is through the degradation of mRNA. If you leave mRNA in your body, it will naturally be degraded.
Why?
Cells contain enzymes that can break down mRNA. If they bump into mRNA, the enzyme will just digest it.
So what do? That defeats the whole purpose of creating the mRNA strand.
You add a poly-A tail to the mRNA. This is just a really long sequence of adenine bases. This protects the mRNA long enough for translation to occur. You can even customize the length of the poly-A tail depending on how much protein you want to make.
Interesting.
The last method of gene regulation I'll be explaining has to do with epigenetics.
What?
This is about changes that occur in the DNA but don't affect the sequence of bases.
How's that possible?
This can occur through methylation, meaning adding a methyl group. Methylation can occur on the DNA itself, often at promoters, or on histones.
What are histones?
They are proteins that DNA wraps around. When methylation occurs on the promoter, this represses gene expression. When methylation occurs on histones, this can make the DNA either wrap more tightly or more loosely to the histones, changing how accessible the genes are. This relates to the concept of epigenesis, the development of an organism from undifferentiated cells. When the cells undergo differentiation, they become specialized for their functions. This is thanks to epigenetic tags like methyl groups, which control which genes are activated. Something that is interesting about epigenetics is that it can be affected by the environment.
Wait, so genes can be affected by the environment?
Yep. For example, there is strong evidence that air pollution is linked to decreased DNA methylation across the genome, which can lead to greater expression of proteins. In particular, when there is more expression of proteins that regulate the immune system, this can cause an increased risk of heart disease, inflammation, and asthma. There is also evidence that things like diet, stress, and trauma can affect the epigenome. Even more interestingly, these tags can be passed on across generations.
So what my parents ate can have an effect on my genes?
In some ways, yeah. When gametes are produced, most of the epigenetic tags are erased, but some remain. So, some epigenetic tags from the parents are passed on to the offspring. This is known as genomic imprinting. The remaining epigenetic tags can have consequences. Do you know what's the offspring of a tiger and a lion?
Oh, was it a liger?
Yeah, that's one of the options, but you can also get a tigon.
Isn't that the same thing?
Nope. A liger has a lion father. A tigon has a lion mother.
Does that make a difference?
Yes, ligers are usually much larger than tigons. This is because different parents carry different imprinting patterns. I encourage you to read into it, it's quite interesting. Going back to the mice. These are twins that have the same genetic information, but their epigenetics are different because the researchers exposed them to different environments. This is why they express different phenotypes.
Oh, okay, now it makes sense.
In general, studies on monozygotic twins are very useful in research.
What do you mean by monozygotic?
It means that they have the same genetic information because they arose from the same zygote, so they are identical twins. There are also twins that don't share the same genetic material because they arose from two zygotes. Monozygotic twins have identical genetic information, so scientists can study them to separate the effects of genes from the environment. Just like the mice. Identical twins, different environments, different outcomes. It's time for shout-out!
This time, the shout-outs go to
And an anonymous fan. If you would like a shout-out, check out the description.